are you guys today? Doing good? Doing great. That's good. What? Yeah, that'll be interesting. Okay. start. Just in case you don't know, my name is Amanda, and this is the biochemistry review for the final exam, specifically for Kokoshkov's class, but if you heard for Kasuli's, hopefully the information will still be relevant. Just a reminder, I am a student. I took this class a year ago, so I do kind of know a lot about the material, but I'm a student, so I'm going to try and cover as much material as we can. But I am liable to make mistakes, so try and make sure you go through and make sure you have the right answers. And we can collectively as a whole decide that, as well as look in the book and stuff to make sure we get them right. As for the multiple choice, so if you don't know, your exam is Wednesday, and it's at 1 p.m. And it's 60 multiple choice. He said it's worth 45% of your grade, and there's 50 points extra credit, right, is what he said? And that'll probably be free response. For the multiple choice, when I asked him, he said to study the same way. So kind of approach it the same way, but now keep in mind as you're going through problems, say to yourself, how could he ask this as a multiple choice instead of as a free response? You can also look at some of the ways he's asked questions in the past. So if you look at, I know your old exams are in more of a free response format, but you can look at those and see kind of what information he thought was important, as well as you can look at the homeworks, the quizzes online and in class, and then any questions in the books. So those are going to be where you're going to find information about what questions he finds important. Yeah, so I kind of have it split out today. I'm going to go through math because that's usually where a lot of people struggle, and then I'm going to quickly go through module 1 and 2 information, and then spend a little bit more time on 3, and then we can do any questions that you guys have. If you have questions while we're going through, and it's relevant to what we're going through, feel free to ask, and we can try and answer that. If we do have to end at 7, so in case there's another review after, so if I don't get to answer your question today, I have a session tomorrow, 12:30 in BA 1218, so you could always come to that. All right, this is a PowerPoint that is up online in the Google Stark site, so you should be able to find it there. There's one slide that I added since I've uploaded it, so I'll point that out when we get there. Okay, so for the math, most of your questions are these. I tried to put down all of the different types of math questions here, so we'll go through each of these that I have written. There may be a few others. Okay, so thermodynamics, these are all the important equations. All of these are kind of from exam 1, except for this one right here, so keep that in mind. This is stuff from our module 3, so we'll go through that. So some sample of problem. He likes the Van Hoff plot, so if you guys remember, this is kind of what the Van Hoff plot looks like. What is your slope going to tell you? What is that equal to when you think about Van Hoff plot? What? Say it a little louder. Negative delta H over R. Does everyone agree that that's going to be your slope? Okay, what about your y-axis? What's that look like? It's going to be your ln KEQ, right? You can see here that's your y-axis. And your x-axis is going to be 1 over temperature. So some ways he's asked this in the past is he's asked you to find delta H or S, which if you remember from the equation on the other side, so you can find delta H and S from this equation. This looks like your y equals mx plus b, right? So this is your slope, as we just pointed out, your y and your x. So you can find your s from the y-intercept, and you can find your delta h from the slope. Another way he asked it, which you guys should have seen on your first exam, is he asked the opposite. He gave you your delta h and delta s, and he said draw the graph. And so from that, you would want to remember that you need to include r in there when you're going to actually plot it. And you can plug in numbers to get your x and y so that you can end up drawing it. Another thermodynamics problem is this one is you're calculating, first you have so delta G naught prime, and then later they ask you to find delta G. So keep in mind the differences between delta G and delta G naught. So your equation that you want to remember is delta G equals delta G naught prime plus RT ln K, right? Now that's the original equation. When it's at equilibrium, your delta G equals 0. And so then that's where you get the equation delta G naught prime equals negative RT ln K, right? Which is another equation you should have seen. So for the first part of the equation, this is what you're going to use to solve for it because it's saying that it's at equilibrium. And so you can take and plug that in. Keep in mind you want to keep in mind the units here as you're plugging this in. Your K is what? Do you guys remember what that equation is? Yeah, products of reactants, which he gave you that equation up there. So that's going to be the concentration of P times the concentration of Q over the concentration of A times the concentration of P. That's where you're going to get your K. And when you plug it in, you want to make sure all your units cancel, right? So you'll have to go through and make sure that all your units match. It may be easiest just to move them all to moles. So if we want to do that real quick, how would 15 millimolar go into moles? Ten to the negative third, 15 moles. And that's going to be, okay, and then you have 40 micromolar. What would that be? 10 to the negative 6. RQ, oh wait, this is backwards, isn't it? All right. RQ is already in molar quantities. And then the last one we have is RP, which is nanomolar would be 10 to the nano 9, right? 5 times 10 to the negative 9. So what do you get when you get that answer? Anyone got it yet? 1.67 times 10 to the negative 3. All right, hopefully someone else got the same answer. All right, we can go with that. So this is your K, and you're going to plug that into this equation. Notice that there's no units now because you've canceled them all out. And so what do you get for delta G when you plug that in? So it'd be delta G naught prime. They'll give you R. RT is, oh, that's a good point. RT is 37 degrees Celsius. Should we plug it straight in? Nope, that's good. We should add 273, so that'll be 310. And then ln of our number. So So what do we get for delta G naught? You guys have it? So here's 60,416. Okay. 16,487. And that would be in joules per mole, right? So that answers the first part of the question, right? Any questions about how to get that? Okay, so the second part of the question, what they've done is they've changed the concentration. So now we're no longer at equilibrium. So we're going to go back to this equation here. So it's important that you remember to go back to that equation, because what you're going to have to do now is add in that value that you just found. So you'll have your delta G and the value that you just found plus your R times your temperature, and then now you have a new K value, right? So again, make sure you check your units when you plug it in. Oh, you guys can't see that. So what will this K be? You have 30 millimolar, right? So that's going to be 30 times 10 to the negative 3 molar. Your B is nanomolar now. 10 times 10 to the negative 9 to make it molar. Your P is now in picomolar. What is that? Negative 12? Or is it negative 15 is femto, right? So pico would be 12. And then the last one is micro. So what do you get that for that value of K? You guys can't see it. Just tell me if you guys can't see it. Well, anyone got it, or do you want to move on? Yeah. Negative 9.83. Okay. So then you would take that value and you would want to plug it into here, and that would give you your delta G, right? So you should be able to do that calculation. Anyone have any questions about that? How could he ask this as a multiple choice? Any thoughts? Just ask it like this and have you do the calculation. Yeah, probably. Okay, so I do have another thermodynamics problem. This one is from module 3. Some of you may have seen it before. I kind of readjusted it. So here, Here what you want to do, you're trying to count how many ATP molecules are required to move so much potassium, and you're moving them out of the cell. Then it tells you the concentrations inside and outside the cell, and the membrane potential and the temperature. and then it tells you how much it is for A2P hydrolysis. With these questions, it's really easy to make a mistake about which direction you're going and what charge or what sign whether it's positive or negative for some of the symbols. So what you need to do is, or what I do is I always draw it out. So if you pretend you have your membrane here, it says you want to move 70 potassium ions out of the cell. So we're moving 70 ions out. So this is in, this is out. And then it says our cytoplasmic concentration, is that going to be inside or outside? Inside, right? So that's going to be 1,000 nanomolar, and the outside is 200 millimolar. They tell you the membrane potential, which is normally represented as delta psi, and you know your temperature is 310. And so the equation that you want to use is found on the other page. It's going to be delta G equals RT ln. If you think of K, then we can show how that works out. And then it's plus ZF delta psi. So if you think about K as products over reactants, right, then you would know that what you want would be, would it be in over out or out over in? Out over in, right? Because then it's going to be, then it will be the products over reactants. So it'll be the concentration of your ions outside over the concentration inside. Now, you have to keep in mind the convention from this one is usually in over out. And so what that means is you have to change, since you've now flipped it, you have to change what sign this is, right? Convention has it as the in over out and it's positive, but now we're flipping it, we're going the opposite direction. So there's a, there's kind of like a rhyme that I found in his book, and it said, if you have X going from out to in, and it's a positive Z value, then that figure, or that end portion should be plus ZF delta psi. But if you have it going X from in to out, and it's a negative Z value, then you want a negative ZF delta psi. So we're moving potassium, right? Potassium has a positive charge, so we're working with positive charge. And we're going from in to out, so we're doing the opposite of this one, which means we want this to have a negative charge, right? According to this. So what we end up with when we plug everything in, we'll have delta G equals 310, and then we have our out over our in, and then we're going to do minus, what's the charge? Plus 1, yeah. He will also give you Faraday's constant. And what's the units for Faraday's constant? Yeah, exactly. I was glad you point that out. So, like she's saying, this is in millimolar and this is in nanomolar. So we'll want to make them the same. How do we do that? What? You just kind of convert it. So you can convert one of them to the other. So this would be, if you want to make them both molar, 200 times 10 to negative 3. And this would be times 10 to the negative 9. Right? Okay. So this was volts per mole per joule, or do I have that backwards? What were the units for Faraday's constant? I think it's joules per volt. And then you're going to times that by, is I right on that? Can you guys see it? No. Okay, so that will be times, now we're going to plug it in as 40 millivolts. No, right, we're going to convert it to volts so that we have it cancel out with this one. So that'll be negative 0.04 volts, right? So some things to keep in mind with this problem, the units, you want to make sure they're canceling out. And then definitely the charge, is it going to be positive or negative here? And that's going to depend on what ion you have, whether it's positively or negative charged ion, and then which direction it's flowing. Does anyone have a value for what this delta G equals? Yeah. 27,584. Okay. Joules per mole. A way you can double check yourself is joules per mole is a unit for delta G, right? So does this answer our question? No? Yeah. For this? Yeah. So this was something I saw in one of his books. So if you're moving the ion from outside of the cell into the cell, and you have a positive charge, so you're moving it into the cell, then it's towards the gradient, so it's, it's going to be positive. Yeah. Well, the delta psi deals with the change in the membrane potential. So this is how the ion's charge is affecting that membrane potential. Yeah, it does say that the membrane potential is constant. Mm-hmm. Oh, no. So I see what you're saying. The membrane potential is what they're saying there, that membrane potential is it's gonna stay here. That's the way it's I'm trying to think. That's the way it's defined. Or that is the change in the membrane potential. The delta psi is negative 40. So it's not asking for what it's, I see what you're saying. It's not, yeah. Yeah, I think that's what it's doing, is it's accounting for how, yeah. Yeah, so if you think about the membrane, you're saying you have a negative inside and a positive outside. So you're pushing a potassium out. And so you're hyperpolarizing it. Okay. Which is why, is that where you're saying it's going to be a negative Z? Right. If you brought a sodium in. Okay. Yeah, that's kind of, I think if you sit down and thought about that's probably like it sounds like you're on the right direction. That's how I was kind of thinking of it. So that's where it's, because of what you're moving it in and versus out is going to affect whether it's positive or negative, yeah. Yeah, so that's where here I had negative Z. So if you moved chlorine, they're saying from in to out, and chlorine has a negative charge. Right. So by putting a negative here, then your negatives cancel out and you have a positive. Yeah. It's a great question. Do you have one? Yeah. So, and that way we don't have to deal with whether this should be positive or negative. We're dealing with this factor. So we'll plug this in as whatever value they gave us, which in this one they said negative 40. And so then we'll adjust for whether it should be positive or negative based on the direction we're moving it, and we'll adjust for that by keeping this in mind. Yes? Hmm? Are you saying? Oh, okay. Yeah, so we've got a negative here and a negative here. So we should get something that's bigger than the two numbers. So what's the? 318. All right. Any other questions? 318? Okay. It will come into play later. Okay, so does this answer the question to the problem? No, right? The problem said how many ATP molecules, right? So what we need to do so we have this delta G tells us how much energy it's going to take to move out 1 mole of potassium ions, right? Well, according to our problem, how many potassium do we actually want to move out? 70, right? So let's account for that first. Let's find out how much energy it takes to move out the potassium, specifically 70 ions. So if we have 35,318 joules per mole, right? How can we get from moles to the number of potassium? Avogadro's number, right? 6.02 times the 23rd. That's going to be ions, or sorry, that's backwards. It'll be 1 mole is equal to 16. And then we can do that times our 70 ions. So our units should cancel out and we should be left with a value in joules. Yes? The only thing is you want to account for the 70. Yeah, you could check and see if you get the same answer. It may still work, so probably. Does anyone have this answer? Times 10 to the what? Negative 18. Okay, so that tells you how much energy it takes to move potassium from inside the cell to outside the cell. So now we want to see how many ATP molecules it will need to move that. And so they told you that there's that each ATP molecule that you hydrolyze, each mole is negative 50 kilojoules. So you can take this amount of energy. Now keep in mind that this one's in kilojoules, so and then we're applying what? Then we're applying Avogadro's number again. So then theoretically, like we said, it'd cancel out, and that would tell us number of molecules. Does anyone have that answer? All right, so any questions about this? Once you pull that out, it'll tell you the number of molecules of ATP, right? So that answers the question at that point. Mm-hmm. He could ask for the number of moles of ATP. He could ask for, how else do you think he could change this question around? Or make it multiple choice? Great, he could ask the opposite. So instead of how many ATP molecules, he gives you the number of ATP molecules and says how many ions can you move. So then you'd have to keep in mind how to work it backwards. Any questions about this before we move on? Does anyone have the answer before I? The 7? Molecules. A lot of ATP. All right, I'm going to go ahead and move on. So that was all thermodynamic stuff. There are other thermodynamics questions, and you can go through the homework stuff to find those, and those were just some of the bigger ones that I saw. Some other math questions he could ask is about the pH of the solution. So keep in mind what's present in the solution. Keep in mind the Ka and Kb, how they are different. And then whether you're working with a weak base and its conjugate acid, when you're working with a weak base and its conjugate acid, or the opposite, you want to use Henderson's Hasselbalch equation. So when I looked at all the problems, he had a lot where he would say calculate the pH of a blank solution. So he could ask where it's a strong acid, or he could ask where it's a weak acid. So if he asks with a strong acid, what happens is you've placed it in solution and now it completely dissolves, right? And so you have no buffering going on, so you don't need to use Henderson-Hasselbalch equation. You can literally just use the equation that pH equals negative log and then your concentration of your hydrogen. And that'll tell you the answer. So keep in mind, all you have to find is that concentration of your H+. If it's a base, then you could find the concentration of OH, and then you know that 14 is going to equal your pH plus your POH, right? Also keep in mind, if it's placed in water and it's in really low concentrations, then your water, what pH does it have when it's just by itself? It's 7, right? So if your pH, if it's in really low concentration, if the acid that you put in is in really low concentrations and your water is overwhelming it, then it may just be at the concentration that the water's at. Like the pH of the solution will just be what the pH of water is. And you should have seen that in exam one. So you can look at that for an example. And with a weak acid, the equation you want to use is now what happens is when you place it in solution, it's going to be going back and forth. It's going to be buffering, right? So sometimes you'll have, so the amount of that's in the acid form and the amount that's in basic form depends on the pKa of the solution. And so that's where you're going to use your Henderson-Hasselbalch equation, right? I think it's base over acid, right? And so here now you're accounting for your base and your acid. Oh wait, for this one, if it's just the pH, yeah, no, that's right, right? That would be the equation you'd want to use. Another way he's asked questions about pH is he'll give you the pH of the solution, but he'll say, what's the concentration of a specific element? And so when you're talking about concentrations, if you think about your K, your Ka is going to have the concentrations of your products over your reactants. And so that's going to come into play, and it's going to help you find, sometimes you have to do you have to find multiple equations, and then, because maybe you don't know all of the numbers there, but then you can use more than one equation to solve for the unknown. And then the other thing is he'll give you like an amino acid. So I think for you guys it was like tyrosine. I think I gave an example with glutamate. And so it has its own pKa, and so it will also do like a buffering type thing. And so he'll say at what portion is protonated versus, or he may say deprotonated, what fraction is it at a specific point in time for this pH. And so the way he had you do that was, he had you do two system equations, right? So you want to use, he had the two equations. The first one was still the Henderson-Hasselbalch. Because when you're looking for a fraction, this would be the fraction that you're looking for, right? That's what you don't know. And then the other equation he had you use was where you have the total amount of your acid plus the total amount. What is that? Will equal a certain amount. And so he usually sets that as 100, so then you can find that percentage later. I have one in my review from the first exam, as well as you can look at his homework questions or at the exam one to see examples. Keep in mind PI and buffering. With the PI, you're looking specifically for where the protein is at 0, right? So where, not necessarily that it doesn't have any positive or negative charges, but that the charges cancel out. And so even if you have a titration curve, it could have like a bunch of different ranges. So the first thing you have to do is find out where your 0 point is. So your 0 point may be all the way up here. And you want to find out, that's going to be at a vertical point, because at that point you only have one species present. And so in order to find out what this pH is, you can use the pKa's above and below it, and you can average them, because it'll be a pH in between those two pKa's. So here we had, this one was 9.78 and this one was 2.35. So if you average those two, you should get 6.07. As for buffering ranges, it's going to be, does anyone remember? Plus or minus 1, yeah. So you're going to have your pKa plus or minus 1. And you'll list all of the different buffering ranges. Don't forget about Beer's law, linking number, percent saturation, and BESI, MS. These are all important equations. For the percent saturation, it could be, here's two different examples, both of which you just plug it into the equation, and then you're solving for one of the variables. For this one, you're solving for P50. For this one, you're solving for YO2. So, yes. Okay. So it's a great question. And if you remember, you should have had one from exam 1. And so what happens is now you have this peptide and it has twice as many. And so it's going to take twice as many twice as much base to neutralize it. So what you actually do is you're extending that region. So if you notice, it starts from 2 and it goes all the way to 4. So you, instead of only taking one unit, so from here we had 0 to 1, and that was 1 fell off. It was finally and then we had here another one, but this one we had 2 histidines, I think, and so it did actually stretch it out. Yeah, if there was 3 or 4, it would stretch it out even more. Does it make sense why? Yeah, because you're adding OH to cancel it out, and you have more present now, so it's going to take more OH to cancel it out, or to deprotonate it. It's a good question. So, yeah, with the percent saturation, just remember the equation, and you should be able to plug it in and then solve for any of the variables that are in that. With linking number, these are just 2 of the different ways you could ask it. With this one, it's all staying in B form, and so they tell you what your W is. It says it's negative supercoils, so they tell you straight away what your W is. Your twist, does anyone remember what twist is equal to? Yeah, base pairs divided by 10.4, since this is bDNA. And then your linking number is just going to be your T plus your W. With this one, you have bDNA, and here it says it's changing from Z to B form. What's the difference between Z and B? What? The W's? Okay. Yeah, the sign of the W, because which one's right-handed and which one's left-handed? B is right-handed? Okay. So B, you're going to have right-handed turns, so that makes your W positive, right? And with Z, you'll have left-handed turns. And so now your W is going to be negative. So you're changing it from Z to B form. He told you how many base pairs per turn for the Z DNA. You should know how many per turn for the B DNA. Now, if you notice here, we're calculating the change in W. And so, did we cut the DNA at all here in this problem? Do you have a question? Oh, is it the other way around? Yeah. Sorry. It could be just the other way around. Yeah. Did we cut the DNA at all in this problem? So if we have a circular strand of DNA and we've changed it from Z DNA to B DNA, it hasn't said anything about it being cut by a topoisomerase or something. So if it hasn't been cut, then our linking number cannot change. So our delta L here is 0. So if you have not cut the DNA, then your linking number, the change in the linking number is 0. Now the linking number itself it will be the same, but it doesn't make sense, the change is 0. Okay, so that's a key point. Now if he does cut it, because there is a problem in your book, I think, where he does cut it, so you can work through that. And then from there, you can calculate your twist. So you'll just do each one. So if you are twisting it from zDNA to bDNA, you can do what the twist is currently, which would be, that's what it is currently, and then you're going to continue twisting it, and that will give you what it is for the B form. 10.4. So you've calculated the twist at what it was, so you have to untwist it, and then, so your twist, you had it twisted up in Z form, and then you have to untwist it, which in this way is a positive direction because you're going from Z to neutral, and then you're going to keep twisting it towards B form, right? Which is why we're adding them. And so then you would end up with your total twist, the total change in twist, which will be, why it's what? So you want to calculate the total amount of change. So it was already twisted up. So you want to untwist it. So that's a X amount of twists, which is going to be 400 over 12. So you're untwisting it, and then you're going to continue twisting it And so that's even more twist that you're adding on to it. Great question. Any other questions about that? Yes. Oh, how do you get W? Yeah, so W can just find by your delta L is going to equal delta T minus delta W. So you can rearrange that equation and find delta W. Yes. Okay, great question. If it was going from B to Z, do you think you would still add them? What do you guys think? Yeah, so if you're going to still add them, because it's the total number of change, so I think you would still probably add them. Yes. I'm sorry, yeah, this should be delta T. Yeah, because so this is the T at your first time point, and then this is the T at your second time point. So then total tells you how much it took to change it. Yeah, sorry, thank you for pointing that out. Yeah, that should be your delta T, yes. What do you guys think? Is T normally negative? I think it is, right? Because twist for, it's the opposite. Yeah. Okay. Okay, I see what you're getting at. So what we're doing though is we're seeing how much it's changed. So we're counting up the total amount of change that it took to go. Like if you think about it, especially because we are twisting it towards bDNA. Does that, yeah, what do you guys think? Would absolute values make sense? So, okay, that works. Does that make sense? If you think about it as absolute value marks. Okay. Any other questions? This is the one slide you guys don't have. So this is an ESI MS one. So the equation that you want to remember here, and it should be on the other slides, is you're going to have your MZ ratio equals your M plus Z over Z. And then you want to find, you have two unknowns here. So they're looking for the molecular weight, which is going to be this M value. That's what you're looking for. But you also don't know your Z. And so in order to do that, you need a second equation, and then you can push the two equations together. So do you guys remember how we do that? Okay, so yes. So Z plus 1 is to the left. Does everyone agree? To the left, or yeah, and then Z minus 1 is to the right. So you can do the same equation where now you've added, so you're saying the 1 to the left is going to be M plus Z plus 1, and then Z plus 1. So you said this is what it is if you added another charge to it, and this is so it would be to the left, right? So when you do this, you want to pick high peaks, and you can pick, you should get the same answer as long as you pick higher peaks. I know one of them, they had peaks like down here, so just stick with the high ones and pick one or two. Pick one of them as your MZ and one of them as your MZ plus 1. So if we said this one is our MZ, so we can solve that for Z, right? And Z would equal Or is it easier to solve for M? It would be easier to solve for M. So M is going to equal 131 over Z minus C, right? Times Z. Is that right? If you rearrange that equation to solve for M, you'll have 1131 times Z minus Z, right? So you can take that value and plug it into your second equation. So for that one, we have 161, so M plus Z plus 1 over Z plus 1. So if we plug in our M value, we'd end up with 131 plus Z. Right? We have a plus Z and a minus Z. And then you can solve for that. Any questions about how to continue this problem? I don't want to take too much time on it, but. So the idea is you set up two equations so that you can solve for M, or you could also solve for Z. Maybe that's the one you don't know. And then you have to push the two equations together to get rid of one of the unknowns so you can find the original unknown that you're looking for. Did I arrange the equation right? There's examples in the homework as well as from the exams. Yes. Yeah, so from here you'll find what Z is. So you'll say Z is equal to some number. But that's not what you're looking for. You're looking for M. And so you can take that value and plug it into here, right, to find what M is. And that will tell you the molecular weight of your protein. So how could he ask this as a multiple choice? He could ask it the same way, but he could also it maybe ask about this information. So that's a concept from that you should know, right? Also remember the theory behind that. Kinetics is from module 2. Remember the different kinds of inhibitors for this graph. What kind of inhibitor is A? Mixed. What about B? Non-competitive? Pure non-competitive? And what about C? Uncompetitive? Non. Okay, and with kinetics, a lot of times he's asking you to find Km and Vmax. With your normal no inhibitor one, how do you find Km and how do you find Vmax? Is A. Is it uncompetitive or mixed? Uncompetitive. Mixed in, okay, so this should be uncompetitive. Are you sure about that? Right. Okay. So uncompetitive is going to be this parallel. We're seeing a change in Km and Vmax, right? That's what you're saying? Pure non-competitive, we have this is our pure. Does everyone agree that that one's pure non-competitive? Yes. There is a slight difference. Who remembers what it is? So. What? Intersects on the. He used it once, but it was very quickly. What do you, say that a question again? So it's a form of non-competitive, but it's a type, but it's very specific because they're saying with the x-axis. Is that what you guys are pointing out? Oh, okay. Yeah. So I was going to put the equations here in just a second. So we'll get there in a second, okay? So the no inhibitor, how do we find Km? One over the x-axis. Does everyone agree? One over the x-intercept. Is it 1 over x or is it negative 1 over x? You guys remember? Is it negative 1 over x? You guys want to look? Does someone want to look that up real quick? I think it's negative 1 over x. Your Vmax is going to be 1 over your y-intercept, right? So your Vmax happens here, which makes sense because that's what your y-axis is. And your Km is going to be on your x-axis, right? So when we're looking this up, so we can find what Km and Vmax is for this one, but then we want to find what it is for each of the other ones. And so that's where we have to take into account the inhibitor that we're using, right? And so the factor that we use to take it into account is alpha, right? So with uncompetitive, what are the equations we need to remember? Does anyone have them? Does anyone remember what chapter this is? 17? Okay. For the sake of time, I'm going to move on, and we can come back to it if we have time. But chapter 17 is going to have all those equations. You could also look at my review where we kind of went through some of them as well. DNA packaging is another math question he could ask. When it's wrapped around the nucleosome, how much smaller is it? By what factor? 7. When it's in the 30 nanometer fiber, how much smaller is it? 8. And when it's then coiled and it's packed all the way, how much smaller is it? 2,000. So these are numbers. He'll give you the amount of base pairs that you have, and you can keep decreasing this. You can decreasing the size from that by using these factors. So module 1 stuff. There could be other math questions. If you guys have questions at the end, we can answer them. I'm going to try and rush through some of this stuff. So module 1, this is your memorization list. Make sure you know it. Kind of treat it like a study guide, so make sure you definitely know everything on those. You should remember it from my exam review for module 1. All of that you can find on the SARD Google site. These are just different ways, things you should know how to do. Find a glycosidic bond, determine whether it's RS, determine whether it's alpha or beta, those type of things. Be able to point out functional groups for all of these different things. And if it's any other mechanisms that are really important. Some other things you should recall. He had 2 mechanisms in the first section. So acid catalyzed hydrolysis, it's going to cut Keep in mind all the different things it can cut. It can cut peptide bonds, ester bonds, phosphoanhydrides. Titration curve, what can absorb at 280 nanometers? Tyrosine and tryptophan, those are the only ones. Polymerase chain reaction, remember there's 3 different temperatures. Does anyone remember what those are? 94, 72. What? Yeah, it kind of varies. Does anyone remember the range? Or 45 to 70? Something like that. Make sure you look that up. Because that was something that he talked about a lot. Keep in mind the reaction that's going on. Also chain termination, remember you had to do with the gel. So you had to be able to put the chain termination, you're using the DDNTPs. And so it's going to stop polymerization at specific points depending on the DNTP that's there. And so that changes how it's going to run in the gel. So kind of remember, hopefully you're recalling all of those things. He's had questions before where he's comparing structures. So he'll give you 2 amino acids and say, why is this one more acidic? Or why does this one have a higher delta G? And so think about resonance, think about steric hindrance, think about things that you've learned from organic. And you can see, I think one of your quizzes had a similar question where you're comparing. So kind of look at those and think about how to apply the information that you've learned to say why these are different slightly. And then also the DNA information, remembering base pairs per turn, etc. Module 2, again, here's your memorization list. Make sure you know everything on here. Keep in mind some of these things are structures. Some of them are, so like here we've got structures. Up here, structures. And then some of these things are mechanisms. These are all mechanisms that you want to know. And then you have a lot of processes here, as well as all of these concepts. Don't forget about those, because that would be a quick multiple choice question, right? So that would be a good one to make sure you know those as well. Some of the overview, when you have a mechanism, there's I think 5 on your memorization list. So make sure you first of all know the mechanism. So maybe he'll put out the mechanism and then he'll label parts like A, B, C, D and say which part is wrong. So then you would have to put, is it A is wrong or B is wrong? So maybe that would be one way he could ask it as a multiple choice. Make sure you can draw the energy diagrams for these, as well as remember when you would do you could put another molecule in to inhibit it. What kind of molecule are you looking for when you want to inhibit an enzyme? It should look similar to something, right? Yeah, so you want it to fit in the binding site, and so specifically you want it should look similar to what it normally puts in there. So it should look similar to the substrate. A lot of times it will look similar to the transition state specifically, right? So you'll want to think about like hydrogen bonding that goes on when you're trying to fit something in, the positive and negative charges. And so you want your inhibitor should look kind of similar to your substrate. And it should look specifically towards that transition state. And so that's where knowing that mechanism will come into play. Also remember in this section he talked about the different classes of enzymes, and so you should know all 6 of these and at least an example for each of those. Remember beta sheets and how to draw them. So with parallel, you've got hydrogen bonding that's kind of back and forth. It's like not straight. But then here with anti-parallel, you're getting straight hydrogen bonding. And you'll also notice that for every amino acid, there's one interacting with another one, and then it skips one, and you have another interaction, and it skips one. And it's the same thing on this side. So those are things to keep in mind when you're drawing them. Also keep in mind, so a parallel, obviously your N-terminuses are going to be on the same side. So when you start to draw it out, but you aren't, when you look at it, so if he gives you two different ones and says which one's parallel, we'll make sure that your N to C is running the same direction. But on this one, you'll have N and C on that side because they're running opposite. And then remember your two types of turns. Which one is more stable? Which one? Type 1 or type 2? Which one? Type 2? Why? It's on the outside? Okay, so do we think that makes it more stable? You're saying it makes it more stable, right? Okay, does everyone agree? What about right here? We look at all those three groups, and what do we notice when we put it in type 2 form? So if you notice, all three are on the same side for type 2, right? So it's cis form, right? So if this is cis and this is trans, so if you think about steric hindrance, which one do you think would be more stable? Yes. 1. So you can double check me on that, but I'm pretty sure you said 1 was more stable because of specifically the R groups here, steric hindrance. So if you have type 2, if you remember, he said you're going to want to have small molecules there, like glycine, because there's only an H. And so, so usually it'll be like small molecules in these two. So maybe he'll ask you something like that on a multiple choice. Like which one's more stable? What kind of amino acid would you see here? What other kind of amino acid may you see in a turn? Proline, right? Proline doesn't make any hydrogen bonding because its H is already occupied. So that would be another way he could talk about that. Some other things to remember from this section. Remember you talked about hemoglobin and myoglobin, just their differences. Where is hemoglobin found? Your blood, right? Where is myoglobin found? In your muscles. Mechanisms of he asked you to memorize the mechanism of the muscle contraction, so keep that in, make sure you review that so that you know it. We talked about antibodies in this section. So what's the difference between a paratope and an epitope? So one of them is on the antigen and one of them is on the antibody. So the paratope is on which one? So paratope is on the antibody, right? Does everyone agree? And the epitope is on the antigen. Remember the peptide fragmentations? Remember he gave you one, it was all fragmented, and he asked you to put it back together, so that could be something he asked. He could also ask about the enzymes that are cleaving. So if you have something then it's ending with a bunch of K's and L's, maybe an R, what kind of, how was it fragmented? What enzyme was used? Trypsin? Does everyone agree? We could say something like that. So recall all of those, make sure you refresh on what they are. Anion, cation, and hydrophobic columns. So with those, just keep in mind, first identify what kind of column it is, so what molecule is going to be attracted to it. So an anion column has anions, that it's going to attract anions, right? That's what it means. So that means it has positive charges. So negative charges will hold on better, positive charges will flow through. So with an anion column, the first thing that's going to elute is anything that's positively charged, right? Then you'll have neutral, and then you'll have negative charges. With cation, it's going to be the opposite. And with hydrophobic, it's attracting hydrophobic molecules, right? So anything with high charges is going to go off first. Anything that has big hydrophobic groups, like alanine, valine, leucine, tryptophan, those ones are going to stay on longer, right? Because the last thing that's going to elute is the one that has more attraction to the column. So when you get to this problem, make sure you identify what kind of column it is, what's going to elute first, what's going to elute last, and then make sure you check what the question is asking. Because he could say order it from first to last, or he could say order it from last to first. And does everyone see how he could ask that as a multiple choice question? Okay. Remember, there's the exhaustive methylation and hydrolysis of polysaccharides, so refresh that. As well as clinical trials. What are the 3 phases? And remember? All right. Yeah. Yes, they're phase 1, 2, and 3. Does anyone remember what is done at each phase? So keep in mind when you go back through it, so phase 1, how many volunteers are there? There's only a few, right? I think it's like 10 or something. 10 to 20? Just 20? 20 to 100? Okay. And so that's how many volunteers you'll have. And specifically here, what are they looking for? Yeah, just like the safety of the drug in general. So here you'll have all of the patients are the same. They're not testing whether it's effective or not. They're just making sure that it's not going to hurt anybody or have any short-term side effects. With the second one, then you're going to be testing whether it works or not against specifically that type of disease. And so you'll have more volunteers. And so then you're testing whether the efficiency of the drug, right? That's where the LD and the other thing are going to come in, right? Your lethal dose, and what was the other one? Effective dose, right? And then you have a ratio, and that gives you your theoretical therapeutic index, yeah. And those are all things that they'll be looking for then. And what about the third phase? What? Long-term effects, okay. You'll have more volunteers. You'll still be testing the efficiency of the drug. Also keep in mind, we talked about drug design and delivery. So the things that you'll want to keep in mind, like all the different barriers you have to go through. So it needs to be kind of hydrophobic, right? Because you want it to go through the membranes of the cells, but then you don't want it too hydrophobic because then it could get stuck in fatty tissue, right? And so thinking about all those different things, there was a good slide on one of his PowerPoints that you could go through. That's from Module 3, though, so keep that in mind. Module 3. So again, memorization list. Make sure you know those. So biological membranes, that's kind of what Module 3 is covering. And if you remember, there's the fluid mosaic model, and then we made a bunch of changes to that. And so we noticed that, yes, it's a fluid mosaic, but it still has a skeleton, right? And that skeleton comes into play with proteins. So sometimes the protein gets caught in an area, sometimes it has free movement, and sometimes it's attached to the membrane. Another update to that was that there's lipid rafts, so there's kind of like these proteins like hang out together and they move together. What about extracellular and intercellular? How do you tell the difference between the two? Sugars, right? Glycosylation. So this is going to be our extracellular and our intercellular. So you'll notice the sugars are up here. He kept asking questions in class about like how things were oriented based on that. So keep in mind if you're seeing sugars attached to something, then it's going to be on the outside of the cell. Also the charge on the membrane potential, so the inside of the cell is going to have a negative charge, right? And the outside will have a positive charge. Okay, so talking about glycosylation, you have N and O linked. So if you notice right here, it pretty much tells you whether it's N or O linked. So the big thing to keep in mind is that O linked is going to have serine and threonine, and N linked is going to have asparagine. And so you should be able to draw those, the ASN and the serine and threonine. And then if he gives you a sugar, then you should know how it attaches. Now if you remember from all of your mechanisms, the most reactive area is going to be that anomeric carbon, right? So it makes sense that it's going to attach there. So it should be relatively easier to recognize now that you've done all the memorization from the beginning, right? You can also have, if you see on this membrane right here, you'll notice that this protein specifically has lipids attached to it, and this allows it to be attached to the membrane. And so this attachment is something he asked you to memorize. So you have these different ones. This is the prenylation, and it's showing the difference between these two is just the number of units. So the farcinolone, I can't say these names. This one has 2, and the GG residue has 3. But it also could be fatty acetylated, and that's where these 2 examples come in. So make sure you have those memorized as well. So then we have the secretory pathway. So if you think about this, the body makes sense when you think about what's going on. So I would recommend for these, just sit down, at least draw it once, so that it makes sense and you understand what's going on and why. So if you think about, he said the lumen matches, does it match the cytoplasm or the extracellular environment? Extracellular, right? Why does that make sense if you think about it? Yeah, so the whole point is you want these proteins to go out. You're secreting them. So it's going to fuse with the membrane and then push out its contents. So it's going to match what it's pushing out into, which is the extracellular environment. And so when you look at this, there's a signaling peptide. So that's going to say, so the ribosome starts synthesizing, and it's like, oh wait, this one needs to go on the outside. So it stops it until the right molecule comes in, and then that can attach it to the rough ER, and then it can continue synthesizing into its extracellular environment, which is equivalent to our lumen. And so it continues synthesizing it, and then you'll notice you have glycosylation going on, right? Which makes sense because it's going to be going into the outside of the cell, and then it'll finish going out. And it can either stay attached to the membrane, or it can fully go in, because if it's going to be secreted, then it wouldn't want to be attached to the membrane. But some will stay attached to the membrane. And so if you sit down and write it out at least once, think about the logic behind it, and it should make sense. How could he ask multiple choice questions about this? What? Yeah, so he could ask about the different steps. So what's going at each point. Any other ideas? Labeling it, okay. Yeah. So then he could also have I talked about the lumen and the extracellular, so maybe something about that. He could also have talking about the glycosylation. What types of proteins are secreted? You guys think of any? How about antibodies, right? Those would be something you would want to secrete. What? Cytokines? Okay. So anything that it wants to talk to other cells with? Oh, you guys can't see that. So that could be another question he asked about it. Any questions about the secretory pathway? Okay, then we have a bunch of different membrane transporters. So the difference between diffusion, passive, and active. Does anyone know? So diffusion and passive, what's the difference? Yeah, so diffusion is just going to go through the membrane by itself. It doesn't need any help. Passive, it doesn't need ATP, but it does need a protein to move it. So whether that's a channel, as you pointed out, or you could also have a carrier like this. So we'll get to that in a second. So diffusion is just going to be going through the membrane. So if you think about that, you're going to need something that's small. So like water is really small. You'll need something that's nonpolar. So benzene could go through because it's hydrophobic. Oxygen could go through, right? And so then passive, it's going to need a protein to help it, but it doesn't need ATP. And then active is going to use ATP. And then you have uniporter, symporter, and antiporter. Which one's which in this diagram? Which one's this one? Uniporter, right? It's only moving one. This one is symporter. It's moving two in the same direction. Sim. And antiporter is going to be this last one. Anti, opposite directions. So as I pointed out, this one is a channel, and this one's going to be a carrier. And so both of these are examples of passive. It just is, it's slightly different. And so with potassium, we'll see examples of both. So valinomycin is an example of a carrier one. So it's going to face one way, pull in potassium, and then flip and face the other way. With all of these transporters, you want to keep in mind why they're selective, especially with the ion movement. So does anyone remember why he said sodium doesn't fit, but potassium does? Yeah, we don't want the sodium to go in. We want this to be specifically towards potassium. And so sodium is actually smaller than potassium. So it has to do with the hydration. And so the way potassium is oriented in here, it fits perfectly when unhydrated, but when sodium tries to go in, it still has, like, it doesn't orient as perfectly. So that's an example of a carrier one. When it comes to channels, so this is one that's specifically for potassium. Does anyone remember why this one is selective for potassium? Hint, it's on this side. So the specific amino acids there, it's TVGYG. And so the way they're oriented helps prevent other molecules from coming in. What kind of transport is this? Active, passive, or diffusion? It's passive, right? No ATP necessarily needed. Is it uniport, symport, or antiport? Are we moving one molecule or two? We're moving one kind of molecule, right? One type, so it's actually going to be uniport. And what's the direction of flow? Inside to outside or outside to inside? Outside to inside? Where is potassium more concentrated? It's more concentrated on the inside, right? And if it's going to be passive, it has to be going with its concentration gradient. So it's going to be going from inside to outside, right? It's going along its gradient, which if you think about action potential and muscles, which we'll get to a little bit later, that will help you remember where potassium, potassium is on the inside and it flows out to kind of repolarize. We'll get to that. But yeah, so potassium is more concentrated inside, and for this example, we're going to be going passive diffusion, so it's going to be moving it out. We also have an example of a potassium channel that's voltage gated. So when the cell, this happens with action potential, right? So with action potential, we had an influx of sodium ions, so now the cell becomes more positive or more negative? More positive, right? And so that's going to affect our S4. And so our S4 is going to have a conformational change, and that's going to open this channel here, and it's going to allow potassium to flow out, right? Because that's the direction of flow. It's concentrated on the inside, and so it's going to want to flow out. So we've polarized the cell, we've made it more positive, and so S4 moves, this opens the channel so positive charges can now flow out, and so then it's repolarizing the cell. And so again, this is still, it's still passive, we still aren't using ATP, and it's still a uniporter. And then there's actually, it's like kind of pulling things in, and there's an inactivation ball, if you remember he was talking about it, and it kind of like once the channel opens up, you get a lot that flows out, but the inactivation ball kind of comes over and it ends up blocking that site, and so that turns off the channels, which is good because you want a huge influx out, but then you want it to stop, like you don't want it to go too far. So that's how it kind of hinders it. Another example of a channel that he talked about was aquaporin. So what are we moving in aquaporin? Just a guess. We're moving water, right? And if you notice here, you'll have RH and Ns, and those are going to kind of affect, it's going to affect that selectivity, right? And if you remember here, he said you aren't actually moving water molecules per se. What you have is you have hydrogen, like the jumping, right? Proton jumping, that's what he called it. So the proton is just jumping through, and so it allows for water to flow. Which way is it going to flow? Inside or outside the cell? Inside? So if you, yeah, so I had it as water going from inside to outside. I feel like it could go either ways depending on what direction you have it. It may be going more outside than inside because of if you think about cellular respiration, so you're forming water in cellular respiration, so maybe that's why you have more of an influx out than inside. This is still, is this passive or active? Passive, right? And is it uniport, symport, or antiport? We're only moving one molecule, right? So it's uniport. Maybe moving a bunch of them, but we're only moving one at a time. We have two examples of glucose. We have two examples of glucose transporters. So this one is sodium dependent. And here's a picture on the right. So is this active or passive transport? Passive? Are you sure it's passive? I think it's actually active. You can double check me on that. Now it does depend on a specific kind of gradient. What gradient is it going to be depending on? If you look at the name, sodium dependent, right? So the concentration of the sodium is what's going to affect the movement of the glucose. So is sodium more concentrated on the outside of the cell or the inside of the cell? On the outside. So if sodium wants to go towards its gradient, then it would be towards the inside. I guess I would make it passive then, wouldn't it? If it's going down its gradient. So maybe it's passive. So the way we have it flowing is we have it going from the, you said sodium is more concentrated on the outside, right? So if we're going sodium from the outside and glucose on the outside, and then we want to move them both into the cell is what we have going on here. Right? So we're moving them both into the cell, you can see up here. So is this uniport, symport, or antiport? It's symport, right? We have two different molecules, but they're going the same direction. Okay, and so we have them coming from the outside, inside. Now what happens is we've pulled it from the stomach, like our lumen, like in our intestines, and we're pulling it into these border cells. And so now from there, we're going to pull it into the capillaries, which is where our other glucose transporter comes in. And so here you can see that one up here. Is it active or passive? Passive. And then it's going to be only moving one type of molecule, right? Glucose, so it's going to be a uniporter. And the direction, if you look at this picture, is going from inside to outside, right? And specifically, these are our border cells. So this is the cells lining the stomach. And it's going to our capillaries. Now something interesting, so this side is our capillaries, if you see. This is where it's going to head. Something interesting, and it has to do with like moving down its concentration gradient. So when the cell moves glucose into when the body moves glucose into the cell, it will immediately phosphorylate it. Why do you guys think that's important? Yeah, so it can't leave. So if it immediately phosphorylates it, now it has a negative charge, so it's not going to go back into our transporter, and it's going to stay in that direction. So it's kind of cool because it's a way for it to move it, a way for it to keep it in the cell, and then also it kind of like tricks the cell, and so it's saying now we don't have a lot of glucose, we just have a lot of glucose 6-phosphate, and so it's like, okay, let's keep moving more in. So yeah, that's kind of cool. Yes. Is it active? So where can we find for sure? In the book? So someone want to look it up real quick? I had originally written down that it was active, so it could be active. If it uses ATP, then that would mean it's active, right? Okay. So even though it would be going down the gradient, if it's using ATP to do it, then it would be considered active. Do you have a question? Or? Oh, okay. So you're saying, because down here, so if you put these two pictures together, you're saying that in order to move the sodium back out, okay, that would make sense. So initially, we're moving it down its gradient, which is why that part is passive, but it's active overall because later on we have to push that sodium back out, and that's through the ATPase, so then that would make it active indirectly. That makes sense to me. Does that answer your question? So let's say it's active. Now that I've changed the answer a couple times. Does everyone see why it's active? Any questions? Yes. GLUT1 would still be passive, right? So its movement isn't. So with this one, it's connected to this one. So the sodium is moving the glucose in, but then the sodium has to go back out because the cell doesn't want to have sodium in there, and so that's why it's costing energy later on. But this one, it's just moving down. That one isn't using ATP, so it'd still be passive. Okay, so here we have, if you look at this picture with the two together, this one we have, this is the sodium-dependent glucose transport. So the glucose is going to use the sodium to get into the cell, but the sodium can't stay there because the concentration, it needs to be on the outside of the cell, otherwise it'll be depolarized and everything. And so it has to be moved back out, which is going to be using the ATPase. And so that one requires energy. But with this one, it's just moving glucose, and there's no other need for another enzyme to so it's not using an ATP. Does that make sense? So that's why we said the first one is active, but the second one was passive. Okay, we also have the sodium-potassium ATPase. So is this one, by the hint, I'll tell you it's ATPase, so it's probably what kind of passive or active? Active. It's going to be moving sodium and potassium, right? So is it symport or antiport? It's antiport, right? And so what's the direction of flow? Does anyone remember that equation? So it's going to be 3 sodium from the inside of the cell and 2 potassium from the outside of the cell. So again, we're going to move them against their concentration gradient, right? So we know sodium is more concentrated on the outside. So we're going to move it against its concentration gradient. Make sure to remember we have ATP in water. And so then it will be flowing to 3 sodium outside and 2 potassium inside. Plus, we have ADP and PI because you used ATP. And this one comes into play with resetting the membrane potential. Then you have lactose permease, and he showed this pretty picture. And so, is it active or passive transport? Does anyone know? All right, does anyone know? Well, okay. So it's active, right? It's going to be using ATP to move in and out, and it's specifically going to be dependent upon this hydrogen concentration. That's going to be what affects how it moves in. So the binding of the H plus is going to move it in, sorry, this way. So the lactose and the hydrogen coming together is going to push it to the other side, and then it loses the lactose and it loses the hydrogen, and it can go back on the other side. So if it helps, draw it out. That's what I would do, draw it out. And then make sure you know those different things about whether it's active or passive. And then the direction of flow, if you notice here, we've got lactose is going from the outside of the cell, and it's using H plus. So then it'll go to the inside. So when on the memorization list, he kind of put all these different ones, and he said the equation was important. So make sure you know the equation, and specifically in versus out, and how it's going, so that because he could have them all look the same, but then whether it's in or out is what's going to be different. So make sure you know which one you're looking for. Another one that I had was the calcium pump. And so specifically, if you look, it's calcium ATPase, so it's going to be active, right? And it's only moving calcium, so it's still uniporter. And the direction of flow, so if you notice, this is your ER, so this is your cytoplasm. And the equation, you can check the slides, but the equation that I found was calcium from the cytosol, and then you're going to be using ATP in water, and it's going to be going to 2 calcium in the ER lumen, and then ADP and PI. Also keep in mind, a way he could ask this is say what kind of, he could ask about what kind of enzyme it is, so like what classification it would be in. Or he could give you just a portion of this and say what are the products. Those are different ways he could make it multiple choice. Yes. A great question. So would it be hydrolase? The different ones? Because it's hydrolyzing ATP. So yeah, I'd probably classify it as hydrolase. And what function is this doing? Does anyone remember where it comes into play? What? Yeah, so it's coming into play with muscle contraction, but specifically it's resetting it. If you look at, which we'll hopefully have time to get to a little bit. Yeah, so this one is actually, if you look further on in my slides, so it's actually right here, and so it's a way to reset it. And so this one is pumping them out, and then this one's going to be pumping them back in. So it'll come. We've talked about action potential a couple of times, but just to make sure everyone knows, he did have this on the memorization list. You'll want to have this diagram. So when it's being depolarized, you have what kind of ion flooding into the cell? Sodium is going to be flooding in. And so sodium is going to rush into the cell. That's our depolarization. Then we have our peak action potential. And then what happens at that point to repolarize the cell? Our potassium channels open, right? If you remember our voltage gated ones. So now potassium is going to flow inside the cell. Outside, right? Our cell is really positively charged. If you look at this, it's been positively charged. It wants to reset, so it's going to flush potassium out of the cell. And then it gets down to the point where it hyperpolarizes. So a little bit too much potassium was pushed out. And then as it's resetting here, that's going to be our sodium-potassium pump. That's going to be pushing it back to get to our optimum membrane potential, right? So make sure you know this diagram. He had a couple, there's 3 different signaling pathways. So if you think about, we have the membrane and we have ways we can push things in and out, but now we want to be able to talk to the outside environment and the inside environment. And so that's where these signaling pathways come into play. So you have something that binds, and it's going to cause changes throughout the cell, and it's going to have an effect. So like this one specifically is going to affect DNA regulate, like what's being expressed in the DNA. And so if you notice, it's called a tyrosine kinase. And so this receptor here has tyrosines on it, and your tyrosines are going to, when your growth factor binds, it's going to cause a conformational change which brings the 2 ends together. So now that they're closer together, they can phosphorylate each other. And so now that they're phosphorylated, they can affect everything else down the way. And then, so it makes the RAS then become, it changes it to the GTP form, right? So this changes, and then this will then phosphorylate down the line. So our primary messenger is what? It's a hormone, yeah. And then this one, he listed it as the growth factor. What are examples of secondary messengers? What? RAS? Okay. Does everyone agree RAS would be a secondary messenger? Did you say RAS or RAF? Okay, and what's a way we can regulate this? He kept talking about ways you want to be able to turn it off, right? What? Yeah, if you dephosphorylate any of these transcription factors, if you want to turn it off quickly, you could go back further up. And so maybe if you dephosphorylate this one, so if you look at SOS here, it's going in the opposite direction. So dephosphorylating would be a way to deactivate it. Another signaling pathway was the CAMP dependent, or CAMP. So With this one and the IP3 one, they have the same beginning part, but then what it affects is different. So with this one, it's specifically affecting our adenylate cyclase. And so when that goes on, then we have, so this one, it comes down. And so a secondary messenger would be CAMP, right? And then this is going on to further activate and inactivate enzymes. What would be, our primary messenger would still be some type of hormone, right? I'm not sure if he listed one specifically. You could check and see. How are ways we can regulate this? What? Yeah, so you can dephosphorylate, you said this one, right? Yeah, so this enzyme it's cutting out, so this is cyclic AMP, and so that's making it just AMP. And so that is hindering the signal, so that would be one way to cut it off. Another would still be, you could switch it from back to, I think in this one it's GDP, and then it goes to GTP, so you could switch it back to GDP. So that would be another way to turn it off. And this heterotrimeric G protein is this one right here. And he just kind of talked about that one a lot, so make sure you know about that. And specifically, it's the alpha portion that moves off and does all the other signaling, right? And then I have some more equations. You guys can look at that on the PowerPoint. Then this is the last signaling pathway. If you notice, we've still got the same thing here, but now we have our, the alpha portion is now affecting phospholipase C. And so, and it's going to cleave this molecule into two portions, and that's going to go off and affect different things. And so, our primary messenger here is still a hormone. What's our secondary messenger? Any ideas? So here we have IP3 and DAG. Those would both be secondary messengers. He gave a list in class. He said the qualities of secondary messengers. They were like, they can be multiplied, they move around the cell. I don't remember all of them, but if you look back on that lecture, I think he had them. And then what are ways we can regulate this? Yeah, so if you push them back together again, then they can't go off and do other things. So with these ones, I would recommend drawing it out at least once. I know it's a lot to draw it out, but then at least you've gone through and said, all right, this is happening at this point because of this, and then specifically look at like the enzymes that he's pointed out in the memorization list. He's like pointed out certain ones, so make sure you know what reaction that is and like the full equation for that. Again, I have more equations, so you guys can look at that at home. With the catabolism diagram, I have an example question here. I don't really have time to go through it today, but if you notice, it's like asking, you know, which part, like how are we making FAD? So if you look here, this is where FAD is being made. FADH2 is being made, so you should be able to count up those two pathways. So you want to memorize this, but keep in mind how everything is moving. So that's where you want to be able to apply the information that you know. If it helps on the test, you can draw it out, and then you can figure out how things are coming. So what products are being formed when, and then like what comes out of, so this is a good one. What is the total reaction of glycolysis? So knowing that one glucose is going to make 2 pyruvate, right? So if you have that equation, you have glucose, and you have 2 NAD plus, and 2 ADP, and 2 PI. Now you want to form ATP, which is why we have ADP and PI, right? So you know you're going to form 2 ATP, and then you will also form 2 NADH an H plus. Sorry, it should be 2H plus. And then you will also form 2 pyruvate and 2 water. So make sure you know that equation. So with all of these, make sure you ask yourself what equation is coming on, so that, because that could be a way he could ask about the enzymes, like what they're doing, so you could ask about the equation. And yeah. And then the last thing I have is talking about substrate level phosphorylation. I think he'll be talking about it tomorrow. This is, he talked about it a little bit, but this is telling you the amount of energy that comes from phosphorylating some of these high energy compounds. And so this was a example question, and you would want to find out the cost for each of these. So you would kind of write out so this one is glucose going to glucose 6-phosphate. So it would be the reverse of this reaction, right? So that's going to be, to calculate the amount of energy, it's going to be plus 13. And then for you have ATP going to ADP. So if you find that on the chart, it's right here. So then that would be plus negative 30, and that would give you your total amount of energy, right? So you have like 2 half reactions, and then you're looking for the reaction of each portion. So find it in the problem. So this one, you know, glucose is going to glucose 6-phosphate. So how much energy does that cost? So it's not giving off energy, it's requiring energy, because you're going the opposite direction. And then here you have ATP being hydrolyzed. So does everyone kind of follow how to do that one? I rushed through that one really quick. Any other quick questions that anyone has? Okay, if you have more questions, I do have a session tomorrow. It's in BA 218. If you have feedback, you can fill out this survey. Otherwise, that's all I have for today.